Hello everyone. Let's move on to the second topic in metabolism of lipids. We will look at oxidation of fatty acids. So we are going to be studying about beta oxidation of saturated fatty acids, unsaturated fatty acids, odd chain fatty acids, alpha oxidation, omega oxidation, and the disorders that are associated with fatty acid oxidation. So before we start oxidation per se, let's look at how are free fatty acids made available in the tissues for oxidation. As you all know, these fatty acids are stored in the form of triacyl glycerols in the adipose tissue. So it has to be released as free fatty acids. So whenever there is action of certain hormones like glucagon or epinephrine. So these hormones will come and bind to the receptor on the cell surface. So when these hormones bind to the receptor, they activate an enzyme called adenyl cyclase, which in turn converts ATP to cyclic AMP. I am sure you have come across this process previously when you did carbohydrate metabolism, that is the regulation of carbohydrate metabolism. You would have seen this cascade happening. So it is the same thing. So ATP is converted to cyclic AMP by adenyl cyclase. This cyclic AMP favors the formation or phosphorylation of the enzyme protein kinase to a phosphorylated form, which is the active form of protein kinase. And as the name suggests, protein kinase, so it is going to phosphorylate other protein. So this protein kinase in turn will phosphorylate the enzyme lipase, hormone sensitive lipase, to convert it to the active form on phosphorylation. Okay, once this lipase is activated, this will act on triacyl glycerol to release free fatty acids. So if insulin is present, then insulin will act via this enzyme phosphatase, which will dephosphorylate the lipase and convert it to an inactive form. So insulin will prevent lipolysis, whereas glucagon will stimulate lipolysis. So now we have seen how fatty acids are made available in the tissue. Before we start oxidation, one small look at the chemistry of these fatty acids. So as you know, there is a carboxyl end and a methyl end for these fatty acids. So from the carboxyl end, the first carbon atom is the alpha carbon atom, and from the second carbon from the carboxyl group is the beta carbon atom, and the methyl group has the omega carbon atom. This is fixed. So starting from carboxyl end, this is going to be fixed. From carboxyl end, first carbon is alpha carbon, second is beta carbon, and the methyl group will be the omega carbon atom. So based on where the oxidation is starting, we can have alpha oxidation, beta oxidation, and omega oxidation. Apart from this, for very long chain fatty acids, we will have peroxisomal fatty acid oxidation. So this is the same numbering system that we looked at in a showing it to you in a linear form. So the principal method, most common oxidation is beta oxidation. Okay, this process starts at the beta carbon atom, that is the second carbon from the carboxyl end, and that is why this is the beta oxidation. And there were many theories that were put forth, but the main theory that is widely accepted is by Frey-Knoop, which was proposed in 1904. So what happens during beta oxidation? This is sequential degradation of fatty acid. So for every for every time the beta oxidation happens, 2 carbon units will be removed in the form of acetyl CoA. So first we have the activation of fatty acid. We will look at the step later. On activation, after activation, beta oxidation happens, and this alpha carbon with the carboxyl group with the activated coenzyme A will now be released as a acetyl CoA molecule, and the remaining fatty acid will now be 2 carbon atoms less than what we started with. So it is always going to be released as acetyl CoA and never as free acetate. Now tissues where beta oxidation will happen. Most commonly beta oxidation occurs in liver, heart, kidney, muscles, lungs, testes, adipose tissue. If you look at tissue wise where does it happen maximally, then cardiac muscles depend more on beta oxidation for their energy purposes. That is, 80% of its energy is derived from fatty acid oxidation and not from glycolysis. So we have looked at tissues where beta oxidation is happening. Now tissues where beta oxidation does not take place, that is the erythrocytes, brain and adrenal medulla. So if you look at within the cell, in which compartment of the cell beta oxidation is happening, this happens in mitochondria. So obviously we will be able to rule out erythrocytes because they do not contain mitochondria. So beta oxidation cannot happen in erythrocytes. Now coming to brain. Brain mostly depends on glycolysis for its energy needs. Suppose the person is in starvation or is in an energy deprived state, then brain will switch over to ketone bodies for energy, but brain will not utilize fatty acids for energy. Multiple reasons have been put forth, one being that probably the entry of these fatty acids across the blood-brain barrier into the brain is a little longer process. So brain cannot depend on these fatty acids is one reason. The other reason that I have come across is that when beta oxidation is happening, there is lot of oxidants that can be produced and released into the tissue. So for this reason again probably beta oxidation does not happen in brain. So brain will depend on glucose, and once glucose is less, then brain switches over to ketone bodies when they are available. Then adrenal medulla, not sure, I haven't been able to find any answer to why beta oxidation is not happening in adrenal medulla, but these are 3 tissues where beta oxidation does not happen. So as I told you, within the cell it is happening in mitochondria, and these fatty acids are going to be present in cytosol. Okay. So we need to transport fatty acids from cytosol into the mitochondria. Short chain fatty acids, medium chain fatty acids, that is up to about 12 carbon chain length, can easily cross the mitochondrial membrane and enter into the mitochondria. There is no problem for it. Whereas those which are 14 and above, chain length of 14 and above, so they require a carrier for their transport into mitochondria. And especially very long chain fatty acids, if it is fatty acids which are more than 16 carbon atoms, then first enters into the peroxisome where part of beta oxidation happens for a few cycles, the chain length is shortened, and then these fatty acids then enter into the mitochondria for oxidation. So the enzymes are present in the mitochondrial membrane and matrix. Again, there are 3 stages in beta oxidation. So if you get a long essay simply as beta oxidation, you should always start from stage 1, stage 2 and stage 3, not just talk about only stage 3 in your answer. Okay. So stage 1 is activation of fatty acids, activation of fatty acid to convert it to acyl CoA. And remember, I have told you previously also, acyl refers to a fatty acid. So activated fatty acid is referred to as acyl CoA. Stage 2 is transport of these fatty acids into the mitochondria. This requires a specialized protein called as carnitine. Requires carnitine as a transporter for fatty acids from cytosol into the mitochondria. This is stage 2 and there are 2 enzymes required for this stage. We will come to it later. Then stage 3 is called as beta oxidation proper or the actual beta oxidation. So let us look at each of these steps. First step is activation of fatty acids. So in the first step, a thioester linkage is formed between fatty acid and a coenzyme A molecule. This is an energy requiring process. Enzyme required is acyl CoA synthetase. Acyl CoA synthetase. These were previously called as thiokinase. And these acyl CoA synthetases, we have multiple isoenzymes or isomers available and each of them are specific to the chain length. So we have short chain acyl CoA synthetases, medium chain acyl CoA synthetases and long chain acyl CoA synthetases. So fatty acid requires energy, so we have ATP and requires coenzyme A. Enzyme is acyl CoA synthetase and this converts the fatty acid to acyl CoA and ATP is converted to AMP and inorganic pyrophosphate is released in this step. So whenever ATP is converted to AMP, you should consider it as being the equivalent of utilizing 2 ATPs. If ATP is converted to ADP, then that is 1 ATP, whereas ATP being converted to AMP, we consider it as utilization of 2 ATPs. So this step requires 2 ATPs, equivalent of 2 ATPs. And the inorganic pyrophosphate that is formed is immediately converted to inorganic phosphate by pyrophosphatase. And the energy given out by this reaction will drive the reaction forward. So that is stage 1, that is activation of fatty acids. Coming to stage 2, that is transport of fatty acids into the mitochondria. Now as I told you, short chain and medium chain fatty acids do not require any transporter. They can be directly transported into the mitochondria. Whereas long chain fatty acids require a transporter. They are impermeable. When we look at this mitochondrial membrane, it is a double layered membrane. So you have a outer membrane and a inner membrane. So these fatty acids are able to enter through the outer membrane but will not be able to cross the inner mitochondrial membrane. So this requires a transporter molecule called as carnitine. So that is why this is also called as carnitine shuttle. So here the activated fatty acid, that is acyl CoA, condenses with carnitine and the coenzyme A is released and it forms acyl carnitine. The enzyme for this step is carnitine acyl transferase if you are not referring to any fatty acid, but if we are referring to palmitic acid, which is the most commonly synthesized fatty acid and which we are taking as an example for oxidation, then it is carnitine palmitoyl transferase or carnitine acyl transferase, CAT or CPT, both of them are correct. And this is CPT1, CPT1 because we also have a CPT2. So now acyl CoA condenses with carnitine, coenzyme A is released and we form acyl carnitine. Next there is a translocase. This translocase will help to transfer this acyl carnitine into the mitochondrial matrix. So as you can see here, CPT1 will convert acyl CoA into acyl carnitine, acyl CoA plus carnitine forming acyl carnitine. So there is a translocase, this is a protein present in the inner mitochondrial membrane which allows acyl carnitine to pass into the mitochondrial membrane, here into the mitochondrial matrix. In the inner mitochondrial membrane we have CPT2, that is carnitine palmitoyl transferase 2 or carnitine acyl transferase 2, which will now re-esterify the fatty acid with coenzyme A and carnitine is released. So acyl carnitine plus coenzyme A will form acyl CoA plus carnitine. This carnitine leaves out through the translocase. So for every acyl carnitine entering into the mitochondria, one carnitine will exit out of the mitochondria, which in turn can bind to another fatty acid to transport it into the mitochondria. So this is the transport of fatty acids from cytosol to the mitochondria. Now suppose there is a disorder of carnitine metabolism. Either the synthesis of carnitine is defective, or the transport of carnitine into the cell is defective, or there is excessive excretion of carnitine. In any of these three conditions, carnitine will be less. So when carnitine is less, the long chain and very long chain fatty acids cannot enter into the mitochondria for oxidation. So this has been seen in preterm infants, sometimes in adults with organic acidurias. So to neutralize this acid, carnitine binds with these organic acids and is excreted as a salt of these organic acids. So there will be more excretion of carnitine. So the main symptoms the person is going to present with would be hypoglycemia, because beta oxidation usually happens after the glycogen stores are reduced, that is the person has been fasting for some time, glucose has been used up, now the glucose is being set aside either for RBCs or for brain. So next option would be to go in for beta oxidation. So when beta oxidation is not happening, then all the cells utilize the glucose and they will land up with hypoglycemia. So treatment here would be, if it is defective biosynthesis or increased excretion, then carnitine administration will be of help. If it is defective transport into the cell, then giving carnitine will not help. There can be an enzyme deficiency also. If it is not carnitine deficiency, either CPT1 or CPT2 deficiency can be seen. Again, symptoms are going to be hypoglycemia and the ketone bodies are going to be low or absent. Normally when there is hypoglycemia and beta oxidation is happening, ketone body synthesis will be happening at the same time. So in these people, because beta oxidation is affected, even ketone body synthesis will not happen. So the clinical features will be hypoglycemia and low ketone bodies. CPT2 deficiency, again if it is the neonatal form, it is very lethal. The baby can die in the next few days after birth, because it cannot sustain the longer duration when the feeds are not happening. So it requires frequent feeds. If the feeds are not given within 2 hours duration, then the baby cannot sustain as its glycogen stores will be depleted. So, leads to respiratory failure, seizures and ultimately death. The infantile form again is also lethal. It's usually death happens around within about a year's age, and exact diagnosis is not usually made because it is assumed to be part of sudden infant death syndrome. Again, symptoms will be the same: hypoglycemia and low ketone bodies. Adult form is usually a milder version. So, the person is able to sustain as long as there are frequent feeds and not doing strenuous exercise or physical activity. So, when they are going for, when this person delays taking food for a longer time or goes in for some physical activity, then the muscles are not getting enough energy, and that is when they present with muscle pain, fatigue, excessive fatigue following exercise. So, here treatment would be frequent feeds and avoid strenuous activity. So, now we looked at stage 1 and 2. Now, let us come to stage 3, that is proper beta oxidation. Now, activation is over, transport of fatty acids is over, and stage 3. Beta oxidation is actually very simple. You just have 4 steps. You just have 4 steps that you need to know, 4 enzymes that you need to know, and this will repeat in a cyclical fashion. So, it is actually very easy compared to some of the other metabolisms that you have studied in carbohydrates. So, first step is oxidation, followed by hydration, again oxidation and dehydrogenation. So, these are the 4 steps: oxidation, hydration, again oxidation and thiolytic cleavage to release acetyl-CoA. Step 1, oxidation. Enzyme is acyl-CoA dehydrogenase, and this again we have isoenzymes which are specific to the chain length. So, in the first step, the acyl-CoA, so if you remember, this is the activated fatty acid. So, carboxyl group is now as C double bond O S CoA, coenzyme A has been added, activated fatty acid, and this is the alpha carbon atom and this is the beta carbon atom. So, we are going to look at what changes happen at the beta carbon atom. So, first step is oxidation, that is removal of hydrogen. Enzyme acyl-CoA dehydrogenase. So, acceptor of this hydrogen will be FAD. I am sure you have finished this coenzyme vitamin from riboflavin. So, FAD is the acceptor of hydrogen. So, it is getting reduced to form FADH2, and the fatty acid is now being converted into an unsaturated fatty acid by the introduction of a double bond. Unsaturated fatty acid, that is, this is a trans double bond. So, trans delta 2 because the double bond is present between the carbon atom 2 and 3. So, trans delta 2 enoyl CoA. This is the first step, that is oxidation. The FADH2 that is formed here will enter into the electron transport chain to give energy. Now, next step is hydration, addition of water, enzyme enoyl CoA hydratase. So, we need to add water. So, if you see here, the hydroxyl group is getting added at the beta carbon atom and hydrogen, single hydrogen is added to the alpha carbon atom. So, the beta carbon atom now has a hydroxyl group. So, we have 3-hydroxy acyl-CoA or beta hydroxy acyl CoA. Next step is again oxidation. Again as beta hydroxy acyl CoA dehydrogenase. Beta hydroxy acyl CoA dehydrogenase. Here the acceptor of hydrogen is NAD. Again coenzyme form of the vitamin niacin. NAD is getting reduced to NADH and H plus, and beta hydroxy, the hydroxyl group is now forming a keto group, is getting oxidized to form a keto group. Okay. In the next step, this alpha carbon atom along with the activated CoA group is going to be released as acetyl CoA. So next enzyme is beta keto thiolase, leads to cleavage of 2 carbon atoms as acetyl CoA, requires another coenzyme A so that the fatty acid is going to remain in the activated form, otherwise it will not be able to undergo these reactions. So at the end of the fourth step we have acetyl CoA, which is a 2 carbon compound, and we have a fatty acid which is now 2 carbons less than what we started with. So if we are looking at palmitoyl CoA, which is a 16 carbon compound, at the end of one cycle we have acetyl CoA plus a 14 carbon fatty acid. So next, this 14 carbon fatty acid will again undergo the same steps of dehydrogenation, hydration, dehydrogenation and thiolysis. One more acetyl CoA will be released. Now we have a 12 carbon fatty acid, which again undergoes the same beta oxidation steps to form 10. So by sequential steps, the fatty acid will lose 2 carbon atoms sequentially, and ultimately in the last step a 4 carbon fatty acid enters into beta oxidation and gets split into 2 acetyl CoAs. That is the end of beta oxidation. So if we start with a 16 carbon compound, it requires 7 cycles of beta oxidation, 7 cycles to be completely oxidized to acetyl CoA. So palmitoyl CoA plus 7 FAD plus 7 NAD, 7 coenzyme A plus 7 water molecules are required to form 8 acetyl CoA, 7 FADH2, 7 NADH and 7 hydrogen atoms are released. Okay. So that was about oxidation, beta oxidation of saturated fatty acids. So one small slide for beta oxidation for unsaturated fatty acids. So if you look at the same palmitoyl CoA, the unsaturated form is palmitoluol CoA, where there is a double bond here, where it has a double bond, and this is in a cis bond. This is a cis bond, not a trans double bond. So these unsaturated fatty acids are can be present in triacyl glycerols, phospholipids, animal origin, plant origin. We have lot of unsaturated fatty acids. So initially the activation, transport, everything is similar to what we came across. Now we have 7 CH2 groups here, after which the double bond is present. So initially it undergoes beta oxidation normally till it reaches this position where it is presented with a cis double bond. So it undergoes 3 cycles, as there are 7 CH2 groups. In 3 cycles they will be released, 6 of them, and we are left with CH2, and then CH, there is a double bond that is present here at the beta position. There is a double bond. This is a cis delta 3 enoyl CoA. So, we have seen that in beta oxidation, if it is trans delta enoyl CoA, it can, the beta oxidation proper can happen, whereas these enzymes will be specific to the type of bond. Cis trans also they will not work if it is different. So, we need to convert this cis to a trans double bond. So, if you see here, this is a trans double bond that was present, whereas we have a cis double bond that is seen here. So, now we need some other enzyme to convert the cis into trans double bond. So, that is a isomerase. This isomerase will now convert the cis into a trans double bond and beta oxidation can continue. Suppose this is instead of just one double bond, if there are multiple double bonds that are present, then apart from isomerase, another reductase is also needed to reduce that to form a saturated fatty acid, and then it will continue. So, this is that one small change that will be required if it is a unsaturated fatty acid. It will require an extra enzyme isomerase to convert cis to trans double bond, and in case of PUFA, apart from isomerase, requires one reductase also. So, that is about beta oxidation of saturated, unsaturated, polyunsaturated fatty acids. We'll look at one slide about inhibitor of acyl CoA dehydrogenase. So, this is a fruit. This is called as ackee fruit, present in Jamaica. This is seen in Jamaica, ackee fruit. So, in this ackee fruit, the unripe ackee fruit, not the ripe ones, unripe ackee fruit contains a toxin called as hypoglycin, which will inhibit action of acyl CoA dehydrogenase. So, if acyl CoA dehydrogenase is inhibited, beta oxidation per se is inhibited. So, if beta oxidation is not happening, you already know what symptoms will be seen. That person is going to present with hypoglycemia and low ketone bodies. So, this is also called as Jamaican vomiting sickness. So, if a person consumes large quantities of these unripe ackee fruits, then they end up with Jamaican vomiting sickness. So, manifestations here will be vomiting, hypoglycemia, will lead to seizures. Seizures because glucose stores are depleted, brain will depend either on glucose or on ketone bodies. Now, there is no glucose and ketone bodies are not being formed because beta oxidation is affected. So, brain gets affected, convulsions can lead to coma and death also. Now, this ackee fruit poisoning is there in all the textbooks. So, as further reading, what I want you to do is probably go and search for paper clippings. Few years back, somewhere I guess in middle India, somewhere Madhya Pradesh or Uttar Pradesh, those regions where there have been many cases reported of lychee poisoning. That is, many children have died after consuming lychee. So, I want you to go read those articles, paper clippings, or read what were the symptoms that were there, and maybe search on PubMed for articles as to what is this lychee poisoning, what does it contain, what does it do. So, we will discuss little more about lychee because all of us, I am sure, consume lychee, and lychee poisoning is something you should be knowing. So, there are some similarities between this ackee fruit poisoning and lychee poisoning. So, please read and come. Now, moving on to the energetics, we have seen that during beta oxidation we form FADH2 and NADH and acetyl CoA. So FADH2 and NADH will enter the electron transport chain directly. Acetyl CoA will be channeled into the TCA cycle and will give energy. So let us look at how much energy are we getting from one fatty acid. So ATP yield through the pathway, that is there are two slides here. First slide is telling you about the old way of calculation, that is suppose FADH2 gives 2 ATP. So the next slide has the calculation for the newer thought, that is FADH2 will give 1.5 ATP and not 2. So one FADH2 will give 2 ATP and one NADH will give 3 ATP. So you are getting 5 ATPs from one beta oxidation. Now if we are looking at palmitic acid, there are going to be 7 such cycles. So 7 into 5 is 35 ATPs are coming only from the beta oxidation per se. Now what about acetyl CoA? 8 acetyl CoAs are produced and this acetyl CoA will enter into TCA cycle to give energy. So one acetyl CoA will give you 12 ATPs. So 8 acetyl CoAs are going to give you 96 ATP. So the total energy that we are getting is 96 plus 35, that is 131 ATP. But remember, we had used 2 ATPs for activation. ATP is converted to AMP is the equivalent of utilizing 2 ATP. So net yield is 129 ATP for one fatty acid, 129 ATP. Same thing through the newer calculations where it is 1.5 ATP for FADH2 and 2.5 for NADH. You get 4 ATPs there. So it is 28 and 10 ATP per acetyl CoA, that is 80. So 80 plus 28 is 108 minus 2, 106 ATP per palmitic acid. That is one palmitic acid will give 106 ATPs. Okay, so with this we come to an end of part 1 of beta oxidation. So in today's session we have looked at how are fatty acids released, that is lipolysis, how are fatty acids being made available for beta oxidation. We looked at the different stages of beta oxidation, that is stage 1 activation to form acyl CoA, stage 2 transport into the mitochondria by carnitine or carnitine shuttle, which has 2 enzymes C81, C82, and then beta oxidation proper containing 4 steps: oxidation, hydration, oxidation and thiolytic cleavage. We also looked at how fatty acid oxidation happens in unsaturated fatty acids and polyunsaturated fatty acids. We looked at Jamaican vomiting sickness and we have looked at the energetics for beta oxidation. So further reading, I have asked you to read about lychee poisoning and maybe even about sudden infant death syndrome. Okay, so I'll see you in class and we'll be having a discussion. I hope all of you go through the material given or at least go through what is there in the textbook so we can have a fruitful discussion in class. Thank you.